Hi, I'm Abigail Chu. I'm going to be a fourth-year student majoring in chemistry and biochemistry. The title of my project is Acceleration Two-Dimensional NMR-based Metabolite Identification by Developing poky extensions. Sounds complicated, right? Well, think of it this way. Have you ever needed to take a painkiller like Tylenol or ibuprofen? Well, all those medicines were studied on humans before it got to you, the general public. You don't want to take anything that has not been thoroughly studied on humans, right? This is metabolomics, which is what my project is on. Metabolomics is the study of metabolites. Metabolites are the chemical compounds used in metabolism. This type of study is commonly used for diagnosing diseases, observing the effects of drugs, and detecting adulteration of food. The two leading methods of identification of metabolites are nuclear magnetic resonance spectroscopy and mass spectrometry. These two methods use different machines to analyze the biochemical pathways. For my project, I chose to focus on NMR method. So why NMR? NMR is more reproducible, meaning you can reuse the sample, but it's not sensitive, meaning that it might not be able to differentiate signals that overlap each other. MS, on the other hand, is more sensitive, but it's not reproducible. I chose NMR because it's a growing field and the sensitivity issues is a problem that is currently being researched on. NMR-based metabolomics focuses on how the compounds are composed and its concentration. One type of NMR is the one-dimensional proton NMR. It measures peak volumes to find the concentration of metabolites. A peak in the spectra would show this presence of a compound. Another type is the two-dimensional proton carbon-13 heteronuclear single quantum coherence NMR. Wow, it's a mouthful. But in essence, it's a type of experiment that helps identify metabolites. The additional dimension allows for better separation between peaks, which is also why I chose to focus on the two-dimensional type. For my project, I am pre-configuring raw HSQC spectra found in biological magnetic resonance bank to create library sets. In other words, I'm making the process of identifying unknown metabolites easier. Scientists would compare their unknown to the standards to find its identity. This process is very long and tedious because there are thousands of standards, so they would use computer software to make that process easier. BMRB is an archive of standard metabolite NMR data that scientists would upload on there. There are 1,171 compounds, including duplicates. I converted the 2RR files found on BMRB to UCSF. 2RR is a type of file compatible with another computer software program called Bruker. By converting it to UCSF, you can use on Poky, the software I used for this project. These two files contain only the fingerprint of the compounds. I created save files that contain not only the fingerprints, but also the declarations. I divided the 1,000 plus compounds into categories and color-coded them. I also adjusted the threshold for viewing level, window size, and set the condition. The spectra on the right is an example of what it would look like if a researcher overlaid standards onto the unknown. The light brown region is glucose. As you can see, since it's right on top of some of the peaks in the unknown sample, this means that there must be glucose in the unknown sample. Another possible match in the sample is HEPs. Since the yellow peaks since the yellow peaks match the known peaks in the spectra, then it's most likely going to be HEPs. Poky is the software program that I used for this project. It was created by Dr. Woong Hee Lee's group. It is a modernized version of another software called NMR-FAM Sparky. Poky is a software suite for multi-dimensional NMR and 3D structure calculation of biomolecules. For a future project, I plan to incorporate the library sets I made into an algorithm that automatically suggests compounds for the identity of the unknown sample and then overlay those suggested ones onto the unknown 2D HSQC spectra for confirmation. I hope you have learned something new through my presentation. Thank you to Dr. Lee and the entire Poky team for all the support and Eureka for this opportunity. Have a good day.